Hi all, myself Sonyamal from First MSc Biology, St. Mary's College, Thrissur. Here in this video, we are going to explain in situ ex situ conservation, keystone species, and measuring biodiversity. So let us first move to in situ ex situ conservation. In situ conservation means conservation of plants and animals in their natural habitat or environment. It involves the conservation of total ecosystem in protected area. Protected area means it's a distinct geographical region identified, managed and preserved for long-term conservation of biodiversity. It also refers to the preservation of genetic resources within evolutionary dynamic ecosystem of their natural environment. A large number of organisms is protected and maintained. Best strategy for long-term protection and biodiversity. Examples of in situ conservation are national park, wildlife sanctuaries, biosphere reserve, natural world heritage site, conservation and community reserves, etc. So let us move to national park. National parks are the area which is set aside for the protection and conservation of outstanding natural flora and fauna. It is for the improvement and better management of wildlife and biodiversity. Hunting, killing, capturing animals, deprivation of habitat are prohibited in national parks. Grazing of livestock is not allowed. Human activity is not permitted inside national park. Only those permitted by Chief Wild Warden of state under provision can enter. Examples for national parks are Erevikulam National Park, Silent Valley, Periyar, etc. Next is wildlife sanctuaries. Wildlife sanctuary is an area set apart for preservation of wildlife under natural condition. Here also killing, hunting is prohibited and the removal or exploitation of wildlife or produced from a sanctuary require the recommendation of State Board for Wildlife. Wildlife sanctuaries example is Bandipur Sanctuary and Periyar Sanctuary. Biosphere reserves. Biosphere reserves are protected area where all biological species are given protection and multiple land use is permitted. And the biosphere reserves are established by UNESCO's Man and Biosphere Reserve Program. It also serves to bring man and nature together to preserve nature and natural resource. Three zones of biosphere are core zone, buffer zone, manipulation zone or transition zone. In core zone, human activity is permitted. In buffer zone, limited human activity is permitted. In manipulation zone, free human activity is permitted. Examples for biosphere reserves are Nilgiri Biosphere Reserve, Nanda Devi Biosphere Reserve, Agasthyamari Biosphere Reserve. Next is World Heritage Site. These are internationally well-known biodiversity area and identified by World Heritage Committee. There are 40 World Heritage Sites in India. They include four major types. Wilderness area. First one is wilderness area. These are large areas and they are protected less stringently than biosphere reserve. Here also provide a natural environment for plants and animals. So we can say that it's a tract or region uncultivated and uninhabited by human beings. Next is natural monuments. Natural monuments means it's selected to protect natural monuments and their surrounding areas. Natural monuments are set aside to protect a specific natural monument which can be landform, seamount, submarine cavern, etc. Example is Pulpit Rock in Norway. Next, species managed areas. Specific areas selected for the conservation of certain habitat or species which require constant protection. Specific areas for the conservation of species which require constant protection. Next is protected landscape and seascapes. Examples are Sundar National Park, Kaziranga National Park, Manas Wildlife Sanctuary. Next in situ conservation is conservation reserves and community reserves. Conservation reserves include the reserve and restricted land occupied by the government to protect wild animals. Whereas the community reserve is defined as privately owned land either by an individual or community. Conservation reserves and community reserves in India are forms denoting protected areas of India which typically act as buffer zones. There are 219 community reserves and more than 100 conservation reserves in India. Next, we are moving to ex situ conservation. Ex situ conservation means offsite conservation of plants and animals outside their natural habitats. This is particularly for endangered species, and examples are zoo, botanical garden, gene bank, pollen bank, germplasm bank, etc. First is seed bank. Seed banks are the stores in which seeds are preserved for conserving plant species. Seeds are dried to bring down their water content below 4% and cooled and maintained at a constant temperature of minus 20 degrees Celsius. Here the seeds can withstand extremely low temperature and highly reduced water content, and such type of seeds are called orthodox seeds. There are more than 50 different kind of seed banks in India, and examples are International Rice Research Institute, Manila. Here in this seed bank there are 90,000 samples of different variety of rice plants are preserved. Another seed bank example is International Maize and Wheat Improvement Center in Mexico. There 12,000 samples of maize and 100,000 samples of wheats are present. Next, we are moving to gene banks. Gene banks are the collection of DNA sample together with a sample of the somatic material of organism. Gene banks preserve the stock of both seeds and vegetation materials. For plant, this is done by vitro storage, freeze getting from plants to stocking the seeds. In the case of animal, it is done by freezing of ecosperm in ecological freezer until further needs. Germplasm bag. What is germplasm? Germplasm means it's a plant part from which new plant can be generated. Germplasm banks are establishment primary concern with the conservation of genetic material which may be lost through genetic erosion. By tissue culture and cryopreservation technique, most plants is maintained in a viable state for long period of time. So, what is cryopreservation? Cryopreservation means the preservation of germplasm at a low temperature, ultra low temperature of minus 196 degrees Celsius. At such low temperature, biological activities cease to take place. Cell division stop, no genetic changes would occur. This is especially important in the conservation of vegetatively propagating crop plants. Tissue culture. Tissue culture means the meristematic cells, or small part of the vegetative tissue, are allowed to grow in a suitable nutrient medium to form a small clumps called calyces. These calyces are then preserved in cooled liquid nitrogen, and this can be taken out and allowed to multiply as and when required. Next topic is keystone species. Keystone species are the species which have low abundance in community but have high significance and disproportionately greater influence on the community or ecosystem as whole. Their presence or abundance play a vital role in controlling the relative abundance of some other species. So we can understand that keystone species are essential one in community. Removal of keystone species seriously disturb functioning of community. It may lead to drastic changes in the abundance of some other species. Existence of keystone species is generally or essentially a local phenomenon, and it can belong to a trophic level, any trophic level that can be a producers or predatory carnivores. Examples for keystone species are fig trees, starfishes, wolf, and alligator. Let us explain how fig trees act as a keystone species. Fig trees is a keystone species in tropical rainforest. Many animals such as monkeys, some birds depends upon fruit availability. So when fruits are scarce, they may push to starvation. Fig trees are unusual in that they can produce fruits all around the year. During extreme scarcity, fig trees provide fruit for survival of fruit-eating animals. So when the removal of total fig trees from tropical rainforest leads to local extinction of some frugivorous animals. Another example is wolves. Wolves are keystone species, and the loss of wolves may lead to abnormal increase in the population of deers and some small herbivores. Next topic is measuring biodiversity. Biodiversity can be measured at many different levels, including genetic level, species, community, and ecosystem. So there are genetic diversity, species diversity, ecosystem diversity, and landscape diversity. First is genetic diversity. Genetic diversity is the diversity or variation of gene within and between species. It implies that the number of species differ widely from each other in their genetic makeup. Each species have several varieties and each variety have its own genetic makeup. Genetic diversity in a community is directly proportional to total number of species. So the community with many species have higher genetic diversity. What is species diversity? Means the diversity within the species or variability of the species in a community. Variety of a rare or unrelated species or dissimilar species in a community or area. It promotes interspecific competition and thereby establishes and maintains community stability. Thus, we can say that species diversity greatly varies between ecosystem and it is highly found in ectons, tropical rainforest, coral reef ecosystem, and species diversity is low in isolated island, desert, and tundras. Species diversity is related to two factors: species richness and species evenness. What is species richness? Species richness or species abundance is the abundance of number of species in a community. It represents total number of species in a community. That is, species diversity directly proportional to species richness. Species richness varies with community, geographical region, and environmental condition. Species evenness or species equitability is the uniformity in the number and distribution of number of species. That means how equally how equally or uniformly the species are present and distributed in the community. So it represents the ratio or proportion between different species or functional group of a community or region. Species evenness is maximum when all species of a community have approximately same number of individuals. That is, all species present in same number. Species diversity and species evenness has inverse relation. So greater species evenness means lesser species diversity. Species diversity index. It represents ratio between number of species and their biomass and productivity with reference to a particular community. That is, ratio between number of species and biomass and productivity. It indicates the relative abundance of the different species of a community. Next, we are moving to diversity index. First one is Shannon-Weaver species diversity. It was formulated by Claude A. Shannon. Shannon Diversity Index is used to measure the overall information provided by the sample community. So, by this diversity index, we can measure overall information provided by sample community. Total information or H represent the entropy of whole community, and it is expressed by Shannon-Weaver equation as follows. Here, Pi is replaced by m by n, where m is the number of individuals of species, I and n is the total number of individuals in sample considered. So we can say that higher value of H is, or the greater would be the species diversity of community. Simpson. Next one is Simpson Diversity Index. Simpson Diversity Index is the statistical method employed for calculating the species diversity of different ecosystems by making use of the relative abundance of different species. The system was formulated by Edward Simpson. That is, by calculating the data regarding number of different species and number of times in each species is found are required for calculating Simpson Diversity Index. Simpson Index is ideal or best suited for relatively small size samples, and the diversity index of a perfectly homogeneous population would be 0, while that of a perfectly heterogeneous population would be 1. What is dominance index? Quantifies the dominance of one of few species in a community. Greater value indicate higher dominance, and the dominance indices are generally negatively correlated with alpha diversity indices. More dominant community are less diverse. Next is the Berger-Parker Index. It expresses the proportional importance of most abundant type. This matrix is highly biased by sample size and richness. It does not make use of all information available from sample. Next diversity is community diversity or ecosystem diversity. This is the diversity in composition of community and ecosystem. It represents variability in the continental or global distribution of community and ecosystem type. There are different kind of habitats and ecosystem on earth. The species living in each habitat or area may be especially adapted to biotic and abiotic environment of that habitat. So biodiversity varies with communities and ecosystem. The biodiversity or species richness of a subset of community is called point diversity. That is, species richness in a specific community is called point diversity. If this point diversity is higher and greater would be the community diversity. There are three types of the community diversity. There are alpha diversity, beta diversity, and gamma diversity. Alpha diversity means diversity within a single community or ecosystem. Beta diversity, diversity between two communities. Gamma diversity, diversity of habitat over a large geographic area. That is, it measures overall diversity within a large region. Next or last one is the landscape diversity. Landscape diversity refers to the diversity within and between landscape with respect to the extent, composition and connectivity of their constituent ecosystem. Landscape is a stretch of land with a different type of landform, habitat and vegetation. Thus, it consists of different type of ecosystem. Landscape diversity is composed of patchy diversity, type diversity, and pattern diversity. So we can say that landscape diversity means diversity within and between landscape with respect to composition and extent of their constituent ecosystem. So, thank you.